I'd like to show you some histology of iron staining in the human brain, which comes from Louise van der Weert's lab in the Netherlands. And in this panel, which is sections of neocortex that have been pearl stained for iron, you see on the far left normal neocortex with very little staining. In the middle you see moderate Alzheimer's disease, and on the right you see severe Alzheimer's disease, and you can see the vivid staining of the amyloid plaques for iron. Now, 25 years ago, the late Mark Smith and George Perry published that neurofibrillary tangles contain high concentrations of iron, and here is an illustration from that paper. Now, we've been investigating the relationship between cortical iron and the natural history of Alzheimer's disease to determine whether or not the iron could be playing a causal role. And this, I'm going to show results from a couple of papers that we've recently published, where we've quantified the iron from post-mortem human brain tissue using this instrument, which is the inductively coupled plasma mass spectrometer. And we've done this in collaboration with the Rush University Medical Center and Memory and Aging Project. And we've analyzed a 645 brain cohort that had been characterized for Alzheimer pathology according to SIRAC criteria and either negative or positive, and of course they've got a clinical history of dementia or not dementia and doesn't necessarily match the pathology, which is something which is well known in Alzheimer's disease. And what I'd like to show you are the results from people who had pathology confirmed Alzheimer's disease. So I have 227 post-mortem human brain samples that have been quantified for iron, and also the other pathologies of Alzheimer's disease to determine the relative contributions. So firstly, I'd like to orientate you to the impact of the tangle burden within the tissue at post-mortem. And in this slide, we see tangle burden expressed in tertiles, either low, medium, or high tertiles. On the y-axis is the change in global cognition, and on the x-axis are years prior to death. In this cohort, cognitive testing was done annually, and we have up to 10 years of antecedent cognitive data. And what we can see is that people with the highest burden of neurofibrillary tangles have got a more accelerated onset of cognitive decline. And this is actually known, but it orientates ourselves that we've got this control within the tissue. But in contrast, the burden of amyloid plaque made no difference. It did not appear to be related to the rate of antecedent cognitive decline. And these pathologies are all adjusted for the contribution of each other. So we're looking at the independent contribution of each pathology. So the independent contribution of iron burden, we see that it has a significant impact. So individual cases with the highest burden of iron at death also had the fastest rate of antecedent cognitive decline. So we conclude that higher iron measured in post-mortem human brain was associated with faster disease progression in the decade prior to death in these cases from the MAP cohort. We see similar results when we assay for brain iron burden using CSF ferritin. And CSF ferritin is one of the analytes that is measured in the ADNI cohort, and we're looking at data from the ADNI cohort here. And we've reported that higher CSF ferritin is associated with faster disease progression, whether it be cognition, brain atrophy, and these data show the fluorodeoxyglucose PET results with the highest ferritin levels associated with a more accelerated deterioration in metabolism. Brain iron can also be imaged by MRI technology, and we've used quantitative susceptibility mapping as one of the modalities. You see on the right a picture of the mean changes within the brain comparing Alzheimer's disease to controls, and the regions in red are those with the highest levels of brain iron compared to the controls, and this is comparing 117 subjects who have been followed up every 1.5 years for over a 6-year interval. And these data from the Australian Imaging Biomarker and Lifestyle Study were published a few years ago. And on the left-hand side, we see the change in cognition measured by Z-score change per year, standard deviation per year in episodic memory, and its relationship to hippocampal iron, hippocampal QSM. So to begin with, in individuals who were PET scanned and found to be amyloid negative, elevated hippocampal iron did not appear to impact negatively upon cognition. But in individuals who were amyloid positive, we found that the hippocampal iron had a strong impact in promoting the rate of cognitive decline over the 6 years of the study. So this is iron which is even within the normal range, is found to impact upon the rate of cognitive deterioration. Now, notably, a few years later, Oscar Hansen's group published a paper which reported the relationship between neurofibrillary tangle burden on PET against the iron burden measured by QSM in the neocortex. And this is data from the Swedish Biofinder 2 study. So previously we had thought that iron was an upstream factor which promoted plaque formation or tangle formation. But because we find that iron acts independently of plaques and tangles in the post-mortem cohort from the Rush group, we think that the iron actually plays a role downstream now in terms of promoting neurodegeneration. And that what matters is that a lower level of iron is associated with less neurodegeneration, or a higher level with more neurodegeneration, and that it can be within the normal range, but the more iron appears to be associated with an adverse outcome. And we hypothesize that ferritosis could be the mechanism of neurodegeneration that's involved here, and I'll explain that as we go along. Importantly, iron levels within the brain rise with normal aging. And this is the first study of its kind over 60 years ago, quantifying the levels of iron in brain over the human lifespan. You can see the iron levels rise rapidly in childhood and plateau in early adulthood, but nevertheless drift upwards with advanced age and continue to rise throughout the human lifespan. This has been corroborated in many studies, especially using imaging, and I'll show a lifespan study of brain iron imaging in the lower panel. It's also been corroborated in mice and rats and other mammalian species that have been investigated. And these papers are just a sampling of the number of papers that have been reported that corroborate that brain iron levels rise with aging. So it seems to be a very highly validated phenomenon. And imaging has been used to actually map the brain iron deposition throughout the human lifespan. This has been corroborated at 7T. And this shows you data from a paper from Peter Nestor's lab, which mapped the regions of iron accumulation in the brain that rise with aging. And the regions that are most elevated are the ones where you see significant atrophy in Alzheimer's disease. So iron can be hazardous through a number of mechanisms. Iron forms the hydroxyl radical, which damages any analyte that it decays next to. Iron can also promote the degradation of the antioxidant transcription factor HIF1 alpha. But we've become intrigued in the possibility that the elevation of brain iron with aging could feed into a form of programmed cell death that was described now 10 years ago, called ferritosis. And ferritosis is a form of altruistic suicide, which is a protection against malignancy and infection. So to drill down a little bit into the mechanism of ferroptosis, this is the core reaction that occurs, where cytoplasmic ferrous iron reacts with polyunsaturated fatty acids in the plasma membrane to form lipid peroxides. And the lipid peroxides are responsible for a radical chain reaction that propagates throughout the membrane of the cell and ruptures the cell membrane, leading to cell death. The signal can actually be propagated through neighboring cells so that there is a wave of death throughout the tissue. Now, there are small molecules that are radical trapping agents that can block the lipid peroxide radical propagation. And a commonly used reference compound is liproxtatin, which has got a low nanomolar efficacy, and vitamin E, which is much weaker and is needed to be used in the high micromolar range. But also iron chelation is another way of blocking ferritosis. And the ferroptotic pathway is regulated through many other facets, which are not illustrated on this slide, but it's quite a complex pathway with many ramifications. But lipid peroxides can generate reactive aldehydes that include 4-hydroxynonenal and malondialdehyde. And these reactive aldehydes can react with species such as proteins, leading to cross-linking. And this has been reported in the literature as potentially contributing to plaque formation, to tangle formation, and Lewy bodies. Glutathione plays a key role in regulating ferroptosis as being the substrate for the checkpoint enzyme glutathione peroxidase 4. Glutathione peroxidase 4 is a selenium-dependent protein, which is the chief mechanism for blocking the radical propagation of lipid peroxides. Experimentally, ferroptosis can be induced with small molecules such as elastin, which lowers cellular glutathione by blocking the uptake of cysteine through the cysteine glutamate antiporter. And RSL3 is used to intoxicate glutathione peroxidase 4. So these two molecules will be using in experiments I'll show you in a little while. Importantly, ferroptosis is not merely caused by iron excess, but iron burden does increase the susceptibility to ferroptosis. And this is illustrated in this experiment where neuronal cells have been exposed to elastin to induce ferroptosis. And you can see the kill curve in black. And when these cells have been co-incubated with a sublethal concentration of iron, you can see the kill curve is shifted markedly to the left. So despite the fact that the iron itself is not causing any toxicity, it potentiates the toxicity that's induced by elastin. Why is the brain so vulnerable to ferroptosis? Well, the brain contains high and dynamic iron concentrations. It contains post-mitotic tissue, and ferroptosis can propagate through a wave of death so that neighboring cells, even if they're non-neuronal cells, can influence the neuronal cell survival. It has high oxygen turnover, which increases the likelihood of lipid peroxidation. It's got a high polyunsaturated fatty acid content, the substrate for ferroptosis, and I've emphasized that there is this age-dependent increase in tissue iron. And the features of Alzheimer's disease that are consistent with ferroptosis include a drop in the long-chain polyunsaturated fatty acids, a decrease in glutathione levels, the involvement of glutathione peroxidase 4, lipid peroxidation, which is a feature that has been very highly reproduced in literature, SAT1 elevation, increased lipoxygenase activity, iron accumulation, as I've mentioned, response to vitamin E, which is a result of a phase 3 clinical trial that I'll mention as we go along. And we're investigating the response to iron chelation in a clinical trial. This is an illustration from a paper from Paul Axelson's lab, which shows an amyloid plaque that has been stained in red for 4-hydroxynonenal, which is a lipid peroxidation product, to illustrate how vividly lipid peroxidation is associated with the proteinopathy of Alzheimer's disease. So we've been investigating the possibility that the genes implicated in Alzheimer's disease are involved with ferroptosis or iron metabolism. This shows you the canonical export pathway for iron from any cell, where iron from the cytoplasm moves to the outside through ferroportin. And ferroportin is in the plasma membrane is the only way that iron can egress from the cell. The iron is then loaded onto transferrin through the activity of ceruloplasmin, and this is how the iron is exported in the extracellular fluid. Now, APP expression is promoted by iron through an iron-responsive element in the 5' UTR of the transcript. And we've been publishing over the last 10 years or so about a mechanism where tau can influence the trafficking of APP along the microtubule pathways to move the APP towards the surface and the plasma membrane, where it stabilizes ferroportin. And by stabilizing ferroportin, it promotes the efflux of iron. So APP knockout mice have got elevated brain iron. More recently, we've published data, and this is led by James Deuce, who's at Merck Sharp and Dohme, where we've studied the impact of mutations that influence the beta secretase processing of APP. So we've compared wild-type APP transfected cells to those transfected with the Italian mutation, which is an alanine to valine mutation in residue 2 of the amyloid sequence. And this is responsible for causing an aggressive form of familial Alzheimer's disease, or substitution at the same residue with threonine, which is the Icelandic mutation, which is protective for Alzheimer's disease. Now, it's known that these mutations influence the processing of A-beta, and that higher levels of A-beta are generated with the Italian mutation and lower levels with the Icelandic mutation, and that's led to a simple hypothesis that the amyloid accumulation is the cause of the toxicity. But we investigated the impact of these mutations on the retention of iron in cells in culture. In this experiment, you see the levels of iron in N2A cells that have been stably transfected with APP695 and then challenged with iron. The dotted line is the baseline. In the black bar, you see the elevation of iron that's induced by the challenge, and this is abolished by co-treatment with a base inhibitor B4. The Icelandic mutation it does not experience such an increase in iron after challenge with iron, but the Italian mutation has got markedly more iron elevation that occurs after challenge, and this is suppressed again by the base inhibitor. So this argues that on top of the influence on amyloid generation, that it's possible that the pathogenesis that's associated with these mutations is consistent with iron retention. We've recently published the impact of presenilin on ferritosis, and this is the work of Mark Greeno and colleagues from my lab, which is online now, on cell death and differentiation. And I'll show you data where presenilin knockout mouse embryonic fibroblasts are shown to be sensitized to ferroptosis. So in this experiment, we intoxicate the cells on the x-axis with RSL3 to induce ferroptosis. In the black line, you see the response of the wild-type fibroblasts, so that's a kill curve. And in the red line, you see the sensitivity of the presenilin double knockout fibroblasts, far more sensitive to ferroptosis. When we test cells that have had wild-type human presenilin 1 transfected in, this nearly normalizes the kill curve towards that of the wild-type fibroblasts. But transfection with familial Alzheimer's disease mutants causes an either incomplete or no protection whatsoever. So these data argue that the familial Alzheimer's disease mutations of presenilin are a loss of function. And this activity is related to gamma secretase activity because if we treat the wild-type fibroblasts with DAPT, with gamma secretase inhibitor, we shift the kill curve to the left. So these cells are more sensitive to ferroptosis. As a control, we looked at the presenilin double knockout cells, and the DAPT has no influence on their survival, and this is because the presenilin double knockouts lack a gamma secretase. So as another control, we checked for the sensitivity of the cells to apoptosis, and we found that the presenilin double knockout fibroblasts are in fact less sensitive to apoptosis, in this case induced by camptothecin. So the sensitivity to ferroptosis induced by loss of presenilin appears to be selective. We looked at fibroblasts that were derived from healthy controls and from a presenilin mutant familial Alzheimer's disease case, and we found that the fibroblasts from the mutant are far more sensitive to ferroptosis, in this case induced by erastin, but also induced by RSL3. We wanted to do kill curves on iPSC cholinergic neurons that are derived from familial cases, but it was not feasible because of the need for high concentrations of antioxidants in the cell cultures in order to generate the cholinergic neurons. But we could measure the selenium levels within the cells. And selenium levels were significantly decreased in presenilin mutant familial Alzheimer's disease neurons. And selenium is needed for the production of glutathione peroxidase 4, and glutathione peroxidase 4 levels were concomitantly decreased in these neurons in the mutants compared to the isogenic controls. And the main uptake pathway for selenium is through LRP8. And LRP8 binds with selenoprotein P to bring selenium into the cell. And here the levels of LRP8 in the neurons from the familial mutants were markedly decreased compared to the isogenic controls. Now it's known that LRP8 expression is induced by the Notch intracellular domain, which is also known to be generated by presenilin gamma secretase activity. And we found that if we silence Notch, that we do indeed get a decrease in both LRP8 and glutathione peroxidase 4. And conversely, if we transfect in the Notch intracellular domain, we get an increase in LRP8 expression in both wild-type and presenilin double knockout cells. So to put this all together, we believe that presenilin is acting at the level of Notch in order to influence ferritosis, and that the pathway looks like this. To begin with, presenilin's cleave Notch to generate the Notch intracellular domain, which is a transcription factor that promotes the production of LRP8. LRP8 is responsible for bringing selenium into the cell through binding selenoprotein P. The selenium is then degraded, the selenoprotein P is then degraded to selenocysteine, which is an amino acid which is needed for the production of the active site of glutathione peroxidase 4, and the glutathione peroxidase 4 is the checkpoint for ferritosis. So that's how we believe presenilins influence ferritosis in an indirect manner. That's already been hypothesized that the loss of presenilin function causes Alzheimer's-like neurodegeneration. And this is the work of Jia Shen and Ray Kelleher. They, for example, have studied presenilin conditional double knockout mice and found that there is an age-dependent atrophy of their brains. And we hypothesize that this atrophy is due to ferritosis, and we're investigating that. So to put this together in a model for the natural history of Alzheimer's disease, we believe that the constant elevation of brain iron with aging, which is a needless accumulation of iron, is responsible for elevating iron to a point where ferritosis becomes inevitable in the neocortex, and that this is responsible for the neurodegeneration component of non-familial Alzheimer's disease. But in the case of presenilin mutations causing familial Alzheimer's disease, ferritosis becomes inevitable at a much earlier age. So we hypothesized that all major Alzheimer's genes converge on iron homeostasis or ferritosis. And most recently, we've reported that apolipoprotein E is the most potent inhibitor of ferritosis yet identified. And this is data from a paper in Molecular Psychiatry, which is currently online. And we see that cells that have been intoxicated with erastin to induce ferritosis can be rescued by liproxtatin, which is the control reference compound, and can also be rescued by lower concentrations of apolipoprotein E. And we believe that the lower levels of apolipoprotein E seen in the E4 carriers are responsible for enabling more ferritosis to occur as a result of carrying that allele. So I've shown you evidence that APP, tau, presenilin, and apolipoprotein E are each involved in iron homeostasis or ferritosis. And because ferritosis is a defense against cancer and infection, we predict that Alzheimer's disease genes will feature in innate immunity and cancer biology. So to finish off, I'd like to have a word about therapeutics. And there have been several reports that treatment with iron chelation can rescue the phenotype of Alzheimer models. Intramuscular desferrioxamine has been reported in a phase 2 clinical trial to improve the outcome in patients with Alzheimer's disease. Vitamin E has been reported to ameliorate cognitive decline in a phase 3 clinical trial. It's one of the few phase 3 clinical trials where there's been a benefit of the treatment in Alzheimer's disease. We were intrigued by the report of David DeVos's lab of a phase 2 clinical trial with deferriprone, an orally bioavailable iron chelator, which improved the deterioration in Parkinson's disease and is currently going through phase 3 testing, which should be reported this year. And as a result, we mounted our own phase 2 clinical trial of deferriprone to treat Alzheimer's disease. This study is going to be completed this year, and unfortunately, because of the pandemic, we couldn't hit the milestone that we wanted in terms of the number of randomized subjects, but we will report what findings we find in this double-blinded study. And thank you very much for your attention, and kol tov.